Hello friends, in today's lecture we will talk about two great scientists and their contributions in the field of microbiology, Edward Jenner and Louis Pasteur. Let us first talk about the contributions of Edward Jenner. Edward Jenner was born in Berkeley, Gloucestershire on 17th May 1749. He was a country physician who was also very interested in various natural sciences, studying bird behavior, human blood, hydrogen and hot air balloons, and geology. His broad interest in the sciences led him to carry out the first scientific studies of immunity to smallpox. Smallpox was a dreaded disease in Europe as it was highly contagious and the mortality rates were as high as 25 to 40%, and those who survived had scars due to the blister-like pustules. However, those who survived also obtained lifelong immunity to the disease. Variolation, the method for the prevention of smallpox by deliberate introduction of material from smallpox pustules into the skin, was quite common then. This generally produced a less severe infection than naturally acquired smallpox, but still induced immunity to it. Though like any other doctor of the time, Edward Jenner carried out variolation to protect his patients from smallpox, he was in search for a more predictable and safer method of protection against the disease. From the early days of his career, Edward Jenner had been intrigued by the country lore which said that people who caught cowpox from their cows could not catch smallpox. Jenner had heard a dairymaid say, "I shall never have smallpox, for I have had cowpox. I shall never have an ugly pock-marked face." In fact, it was a common belief that dairymaids were in some way protected from smallpox. This and his own experience of variolation as a boy and the risks that accompanied it led him to undertake the most important research of his life. Cowpox is a mild viral infection of cows. It causes a few pocks on their udders but little discomfort. Milkmaids occasionally caught cowpox from their cows. In May 1796, a dairymaid, Sarah Nelms, consulted Jenner about a rash on her hand. This figure shows Sarah Nelms' hand with rashes as drawn by Jenner in his casebook. Jenner diagnosed cowpox rather than smallpox, as you can see in this handwritten comment made by Jenner. Sarah confirmed that one of her cows, a Gloucester cow called Blossom, had recently had cowpox. Edward Jenner realized that this was his opportunity to test the protective properties of cowpox by giving it to someone who had not yet suffered from smallpox. He chose James Phipps, the 8-year-old son of his gardener. On 14th May 1796, he made a few scratches on one of James' arms and rubbed into them some material from one of the pocks on Sarah's hand. A few days later, James became mildly ill with cowpox, but was well again in a week later. This proved to Jenner that cowpox could pass from person to person as well as from cow to person. The next step now was to test whether the cowpox would protect James from smallpox. On 1st July, Jenner injected the boy with material from smallpox lesions. As Jenner anticipated, and undoubtedly to his great relief, James did not develop smallpox either on this occasion or on the many other subsequent ones when his immunity was tested again. It is important to stress here that the nature of these diseases and their causative organisms, that is viruses, would not be known for over 100 years. Jenner was actually much ahead of his time. Jenner followed up this experiment with many others. In 1797, Jenner sent a short communication to the Royal Society describing his experiment and observations. However, the paper was rejected. Jenner's newly proven technique for protecting people from smallpox was not received well with the medical fraternity. However, what he had discovered could not be denied, and eventually his discovery had to be accepted, a discovery that was to change the world. So successful was Jenner's discovery that in 1840 the government of the day banned any other treatment for smallpox other than Jenner's treatment. Edward Jenner spent much of the rest of his life supplying cowpox material to others around the world and discussing related scientific matters. He was so involved in corresponding about smallpox that he called himself the vaccine clerk to the world. In 1967, the World Health Organization launched its campaign to eradicate smallpox worldwide. They estimated at that time that there were still up to 15 million cases of smallpox each year. The biggest problem areas were South America, Africa, and the Indian subcontinent. Their approach was to vaccinate every person in the areas at risk. Teams of vaccinators from all over the world journeyed to the remotest of communities. After a period of watching for new cases, in 1980, the WHO formally declared smallpox is dead. This is the WHO certification of smallpox eradication in 1980. The most feared disease of all time had been eradicated, fulfilling a prediction that Edward Jenner had made in 1801. It has been estimated that the task he started has led to saving of more human lives than the work of any other person. The last remaining specimens of smallpox virus are now held in just 2 laboratories in Siberia and USA. The samples used for research are afforded higher security than a nuclear bomb. One day, they too will be destroyed. Smallpox has become the first major infectious disease to be wiped away from the face of the earth. Let us now discuss the contributions of Louis Pasteur in the field of microbiology. Louis Pasteur was born in 1822 to a sergeant in Napoleon's army. He was a professor of chemistry at the University of Lille, France, and started his memorable scientific career by studying the shapes of crystals. While working on tartaric acid crystals, he proved that organic molecules with same chemical composition can have different structures. They are mirror images of each other, as can be seen in this picture, and are called stereoisomers. With this work, he launched the new science of stereochemistry. Pasteur was the most powerful opponent of spontaneous generation theory. He firmly believed that microorganisms were present in the air. He passed air through cotton fibers. The fibers stopped solid particles. He dissolved the cotton in a mixture of alcohol and ether. The particles trapped in the fibers fell to the bottom of the liquid and were examined under microscope. Pasteur found that they were similar to the microorganisms found in putrefying meat. He concluded that these microbes are constantly present in the air and are being deposited on all objects. Let us now move on to the contributions of Pasteur in the field of fermentation. The chemists of that period believed that conversion of sugar to alcohol and carbon dioxide was a simple chemical process. Ironically, it was Pasteur, a chemist, who convinced the world that all fermentations are a result of microbial activity. In those days, winemaking was an important industry in France. The distillers were worried because the quality of their wine was deteriorating. In the summer of 1856, M. Bigot, father of one of Pasteur's students in chemistry, called upon Pasteur to help him overcome difficulties he was having manufacturing alcohol by fermentation. Pasteur investigated the fermentation process and found that their problems were due to conversion of sugar to lactic acid by rod-shaped bacteria. His microscopic observations made him realize that when alcohol was produced normally, the yeast cells were plump and budding. But when lactic acid would form instead of alcohol, small rod-like microbes were always mixed with the yeast cells. He found that yeast characteristic of the alcoholic fermentation had been replaced by bacteria which brought about lactic fermentation, which made the wines sour and bitter. Pasteur then developed a process of heating the grape juice just enough to kill most contaminating bacteria without changing the juice's basic qualities so that it could be then inoculated with yeast to ensure that alcoholic fermentation would occur. Today, pasteurization, as this process is called, is routinely used in fermentations and dairy industries. During his studies on butyric fermentations, Pasteur discovered another fundamental biological phenomena, anaerobic form of life. He microscopically observed the fluids that were undergoing butyric fermentations. He observed that bacteria at the margin of the drop of fluid in contact with air became immotile, whereas the bacteria in the center of the drop remained motile. The suggestion that air might have an inhibitory effect on microorganisms was confirmed by Pasteur, who showed that when air was passed to the fermenting fluids, it not only retarded but completely stopped the fermentation. He introduced the terms aerobic to designate life in presence of oxygen and anaerobic, life in absence of oxygen. If we go back in history, during the latter part of the 17th century, it was the Dutch scientist Anton van Leeuwenhoek who first observed the existence of anaerobic organisms, or those organisms that can live and grow without the presence of oxygen. Pasteur thus rediscovered anaerobiosis around 200 years after Leeuwenhoek. His rediscovery led to the establishment of the concept of aerobic and anaerobic life. This process of inhibition of the fermentation process by oxygen came to be known as the Pasteur effect. In the middle of the 19th century, the French silkworm industry was being destroyed by infectious diseases which were killing a great number of silkworms. In 1865, Louis Pasteur accepted the request of the French government to investigate pebrine, a silkworm disease, though he knew nothing about silkworms. For several years, Pasteur struggled but eventually succeeded in isolating the parasite causing the disease. The parasite was found to be a protozoan. After several years of research, Pasteur was able to save the silkworm industry by developing a method of preventing contamination of healthy silkworm eggs. The method was soon employed by silk producers all over the world and is still used in silk producing countries. After silk, Pasteur turned to wool and tackled the problem of anthrax, a disease of cattle. The causative agent of anthrax had already been identified by the time Pasteur entered the arena. It was none other than the great German physician Robert Koch who had isolated the anthrax bacillus. However, definitive proof that it was the isolated bacillus and not any other agent that was carried in the original medium that was responsible for causing the disease was still lacking. Pasteur inoculated a drop of infected blood from a sheep dying from anthrax into 50 ml medium and incubated it, and repeated the process of dilution 100 times. Thus, the original sample was so highly diluted that there was no chance of any molecule from the original sample remaining in the culture. Yet, the last diluted culture was as active as the first in producing anthrax, as only the bacillus itself, by growing each time in the new culture, could escape dilution. It proved beyond doubt that the anthrax bacillus and nothing else could be responsible for the disease. Pasteur continued to make discoveries concerning the cause and prevention of infectious diseases. Louis Pasteur conducted numerous experiments in his career to demonstrate the relationship between germs and diseases. He established without doubt that certain diseases are caused by microorganisms. Hence, though he was not the first to propose it, Pasteur is credited for his work on the germ theory of disease. Let us move on now to Pasteur's contribution in the field of vaccination. Louis Pasteur's first important discovery in the study of vaccination came in 1879 and was regarding the disease known as chicken cholera. Around 1880, Pasteur isolated the bacteria responsible for chicken cholera and grew it as a pure culture. Pasteur arranged for a public demonstration of an experiment which was successful in many trials. He inoculated healthy chickens with his pure cultures. Students, what would you expect to happen? Right. Even Pasteur anticipated and expected the chickens to die. But to his surprise, his chickens remained healthy. Pasteur realized that he had accidentally used cultures which were several weeks old instead of fresh cultures. Some weeks later, Pasteur repeated the same experiment. this time using two different groups of chicks. One of the groups had been injected at the first demonstration with the old culture, whereas the other group had not been exposed to any culture previously. Both groups were now injected with fresh chicken cholera pure cultures. This time, the chicks of the second group became sick and died, whereas those of the first group remained hale and hearty. Students, can you guess the reason for these results? Pasteur too was puzzled at the results, but he soon found out the reason. In some way, the old cultures lost their ability to cause disease. But these attenuated bacteria still retained their capacity to produce antibodies in the host. Pasteur realized that in a sense he was repeating the studies of Edward Jenner, who had conferred immunity to smallpox by vaccinating individuals with a mild form of cowpox. He called the attenuated cultures as vaccines and developed the concept of vaccination, acknowledging Jenner's work because in Latin, vacca means cow, and we have studied about Jenner's work on cowpox and smallpox. By now, Pasteur had become very famous. It was a common belief that he could work wonders with bacteria and control any infection. Although the method of vaccination had been previously developed by English physician Edward Jenner in 1796, the difference between smallpox vaccination of Jenner and Pasteur's chicken cholera vaccination was that the weakened form of the latter disease organisms had been generated artificially. Thus, it was not required to find a naturally weak form of the disease organism. It was this discovery that revolutionized work in the prevention of infectious diseases and has helped save countless lives since then. The final and certainly most famous success of Pasteur's research was the development of a vaccine against rabies or hydrophobia. This was the first human disease he was working on and was a challenge for him considering that he was not a physician by profession and was actually a chemist. After a lot of hard work, Pasteur had succeeded in producing a vaccine against rabies. Using a concoction of dried spinal cord material harvested from rabbits, he had saved the lives of 40 rabies infected dogs, but was yet to test it on humans. Pasteur was not very sure if he was ready for human trials, particularly because he was not successful in isolating the rabic substance. But on July 6, 1885, a 9-year-old boy, Joseph Pasteur, and his mother appeared at Pasteur's laboratory. 2 days earlier, the young boy had been bitten repeatedly by a rabbit dog. His mother knew that her son would die, and hence she appealed to Pasteur to treat her son because she wanted to take that 1 in a million chance which could probably save her son's life. Pasteur administered the vaccine and was surprised and relieved when Joseph Pasteur survived and recovered from rabies. Rabies was the last major research of the master scientist. His health was failing, and paralysis of his left side from a serious stroke he suffered in his 46th year made his working in the laboratory increasingly difficult. Pasteur died on September 28, 1895, after suffering additional strokes. France showed its gratitude to Pasteur by establishing the Pasteur Institute in Paris. Today, Pasteur Institute is a thriving research center, a true tribute to its founder. Shortly after his death, the London Illustrated News published a full-page portrait of Pasteur on its front page as a tribute to the great scientist Louis Pasteur.